Welcome back, everyone. I'm Hossein Borhan, and a research scientist in Agriculture and Agri-Food Canada here in Saskatoon. I'm your chair for this afternoon session. The session will examine strategies for canola disease management, especially insight from plant-microbe interaction. So we have 4 speakers for this afternoon sessions, and we have a few minutes after each talk for questions after each talks. So please ensure that the presenters are staying with the allocated time, and we have time for questions after the talk. The first speaker is Darrell Desveaux. He is a professor at the Department of Cell and System Biology at the University of Toronto, and he will discuss the immunity landscape of oilseed crops. Darrell.
Thanks. All right. Okay. I guess everybody can hear me. Thank you very much. Thanks, Hossein. Thank you to the organizers for inviting me to give our talk about what we're doing in oilseed crops. We're really excited because we were working on Arabidopsis for quite a long time, and over the last 4 or 5 years, we've transitioned what we've learned in Arabidopsis to oilseed crops, and more specifically, Camelina sativa, or false flax, and canola. So I work on the Pseudomonas syringae, which is a gram-negative bacterial pathogen of plants, and it can pretty much infect any plant that you eat or that we grow in agriculture, and that includes Camelina sativa, and I'll show you in later slides that it also infects canola, and it infects and causes disease on these plants using a needle-like structure that's shown in the middle of this slide called the type 3 secretion system. And the type 3 secretion system isn't really, it's like a needle that injects virulence proteins into host cells in order to cause disease, to promote a pathogenic life cycle. And it requires this type 3 secretion system to cause disease. If you knock it out, this Pseudomonas syringae is not virulent on any of these hosts. So although Pseudomonas syringae is very broad spectrum in its ability to cause plants, it's subdivided into what are called pathivars. These are like strains that are host restricted to individual types of plants. And each one of these strains or pathivars has a unique suite of type 3 effectors, typically between 20 to 40 effectors, these virulence proteins, that are injected into host cells in order to cause disease on that specific host. So it's thought that, for example, this the Glycinia pathivar, the strain that causes disease on soybean, is thought to have a suite of effectors that's required to cause disease on soybean. So disease is very prominent and can be very agriculturally and economically important, but it is the exception because plants have come equipped with a very effective immune system. And that immune system can be simply divided in 2 layers of pathogen recognition. There's an extracellular layer on the outside of plant cells where the plant cell can recognize microbial body parts. And these are like bacterial flagella, for example. And that mounts a first layer of immunity called PTI or PRR-triggered immunity. It's not too important what the name is, but it's a broad spectrum immunity. It recognizes conserved microbial body parts, and it's able to protect the plant against most microbes that would like to, live in a plant environment. But most pathogens have come equipped with these effectors, and they deliver them using different mechanisms. I talked about the type 3 secretion system, the needle, but insect pathogens, fungal pathogens, oomycete pathogens all have their own ways of injecting effectors into host plants. And one of their major functions is to block that basal level of immunity. So they can suppress that layer very effectively and allow pathogens to cause disease inside the plant. But plants have a second layer of immunity inside the cell, and that's the recognition of these effectors by resistance proteins. And these resistance proteins, which most of you who've worked on immunity or disease in plants, you know about these resistance proteins. They induce a very strong immune response, and it's been typically referred to as gene-for-gene resistance. It's also referred to as effector-triggered immunity, and it's mediated by this resistance proteins inside the plant cell These are nucleotide-binding, leucine-rich repeat proteins that recognize effectors and then mount a very strong, amplified immune response that protects plants against pathogens. Because there's something wrong. If there's a pathogen that's injected an effector into a plant cell, it's time to take this seriously and kill off that pathogen. And that's what effector-triggered immunity does, or gene-for-gene resistance. So it was well known that this PTI, the first level of extracellular immunity, is quite broad spectrum. It can protect the host against most pathogens because it recognizes conserved molecules. But gene-for-gene resistance was always thought to be more niche, right? You needed a specific gene on the pathogen side to be recognized by a specific gene on the plant side. And how broad spectrum can such a niche resistance be, especially considering that, every pathogen out there contains between 20 to 100 effectors or even more, and they all have different effectors, right? So how good is effector-triggered immunity at protecting plants against a broad swath of pathogens? Whoops, what did I do? Blank. All right. So the real question was, to what extent does effector-triggered immunity, gene-for-gene resistance, protect a plant host against all the effector diversity that's found in a pathogen, and in our case was Pseudomonas syringae. So in order to do this, we wanted to identify all the effectors that we could in Pseudomonas syringae, and we're lucky because effectors in Pseudomonas syringae have been very well characterized. So the lab of David Gutman had fully sequenced over 500 strains of Pseudomonas syringae from over 100 different hosts. And then from those strains, we could extract all the effector sequences, and there was over 5,000 unique effectors in Pseudomonas syringae. Now that's still a pretty high number if we want to do functional work. So within these 5,000, we could subcategorize those into 70 related families. These are evolutionarily related families of effectors. And we could further subdivide those families into subgroups. And those subgroups are highly related subclades that I've indicated there with the arrow, the red arrows. And these subclades are very highly related. And we actually said that they have to be at least 95% identical to every other effector within this subclade. And then we could choose one representative from each one of these subclades to then basically build what we call the Pseudomonas syringae type 3 effector compendium of 530 effectors that represents basically the diversity of effectors that's found in this one pathogen. All right, so that's 530 virulence proteins that basically simplified represents all the virulence proteins in Pseudomonas syringae. And then we wanted to ask, how good is a host at recognizing this diversity, these 530 effectors? So we had them all synthesized with their native promoters. Then we put them into one Pseudomonas syringae strain that's virulent on the model host, Arabidopsis thaliana, related to canola. And then what we could do is then do spray inoculations on Arabidopsis and look for resistance. And this is what that looks like. So normally, if you spray Arabidopsis plants with PTO DC3000, that's the Pseudomonas syringae strain that we're using, it'll kill the plant over about 5 to 6 days. But if you have an effector that's recognized in a gene-for-gene manner in the host side, it'll protect the plant. As you can see for the HOP-F2A example here, those plants stay green because of the effector-triggered immune response. So then we could say, okay, now let's screen all our 530 effectors and ask, how many does Arabidopsis recognize? So this was the work of one grad student, Bradley Laflamme, along with some other members of the lab. He took all the 530 effectors in our diversity suite and screened them all on Arabidopsis. And then he could identify a lot of immunity. And rather than doing this qualitatively, we could also quantify the amount of immunity just by simply measuring the number of green to yellow pixels. Green being resistant, yellow being susceptible, more virulent. Those are the disease symptoms. And then we could set a cutoff and identify those that triggered an immune response. And to summarize, what we found was that in this ecotype, this one type of Arabidopsis, was able to recognize 19 of the 70 effector families found in Pseudomonas syringae. That's over a quarter of the effector diversity that's found in Pseudomonas syringae. So that was pretty impressive. This added a lot new families that we didn't know were recognized by Arabidopsis. But what was even more impressive is when we mapped these immune-eliciting effectors back onto the syringae phylogeny, the pathogen phylogeny. So what I've indicated here at the bottom is the phylogeny of Pseudomonas syringae, and each one of the dark blue bars here represents the presence of an effector that elicits an immune response in one of those strains. And what should immediately come out is that most strains contain an immune-eliciting effector. And at the top, I've just collapsed everything into, does it contain any elicitor? All right, any immune elicitor. And the take-home from this is that 97% of Pseudomonas syringae strains have at least one immune elicitor in Arabidopsis, all right? And in fact, there's 71% of the strains have multiple immune elicitors that would be recognized in Arabidopsis, meaning that, yes, this level, gene-for-gene resistance, is quite pervasive and can be often multi-tiered against a pathogen. So yes, it can provide broad-spectrum immunity. If you look at one pathogen like Pseudomonas syringae, almost every strain has an immune elicitor, right? So it can provide broad-spectrum immunity in addition to that strong niche immunity that's being bred into crops. When we use the flip side and we ask, okay, well, how many resistance proteins are actually required to confer this type of immunity against Pseudomonas syringae? Well, we can map the known resistance proteins that are known to recognize all of these immune elicitors in Arabidopsis. And we see that there's 8 resistance genes that are able to confer pretty much a blanket immunity against Pseudomonas syringae. These 8 resistance proteins that I've listed here on the left can basically recognize 97% of strains. And if you look at the bottom two there, this ZAR1 and CAR1, they can recognize 95% of strains. So 2 resistance proteins could basically confer broad-spectrum immunity against Pseudomonas syringae in Arabidopsis. How do 2 resistance proteins recognize so many strains with such diversity of effectors in them? They use 2 different strategies. This CAR1 at the bottom basically recognizes a group of conserved effectors, right? So like those extracellular receptors that recognize conserved body parts, this resistance protein recognizes conserved effectors, right? So that way it can recognize many different strains. ZAR1, on the other hand, recognizes effectors indirectly through the manipulation of a kinase network. So what I've shown there is the kinase network that's manipulated by effectors. And these are normally manipulated in order to cause disease. But what ZAR1, the resistance protein, does is it monitors this kinase network for any perturbations by the pathogen. And once the pathogen perturbs that kinase network, that's a trigger for ZAR1 to signal. And it turns out there's a lot of effectors that want to target that kinase network. There's 5 in Pseudomonas syringae and at least 1 in another Xanthomonas bacterial pathogen of plants. They all target this kinase network, and as soon as they touch that kinase network and modify it in some way, that activates the ZAR1 resistance gene. So that way, ZAR1 can recognize many strains simply not by monitoring for effectors, but by just closely monitoring for perturbations of itself. Anything that attacks the kinase network that's so precious, I'll trigger the immune response. So I'm going to get back into ZAR1 because it's also conserved in canola and in camelina. So what about crop plants? This is all done in Arabidopsis thaliana, the model plant that's very powerful for studying all of these immune responses. But does this translate into crop plants? So we started translating this work into oilseed crops, camelina, and canola. And they're all part of the same family of plants, the Brassicaceae family of plants. So we chose camelina, which is false flax, and Brassica napus, which is canola, obviously. And they're evolutionarily distant from Arabidopsis. Camelina being more closely related, diverged about 8 million years ago from Arabidopsis, the wild relative. And canola is more distantly related to Arabidopsis. About 23 million years ago, it diverged away. But this presents a very nice continuum a gradient of relationship, right? Evolutionary distance from Arabidopsis as well as domestication. We can think of Arabidopsis as the wild version of a plant, right? As maybe not true anymore because they're all cultivated in the lab for so many years. But it is relative to Camelina and Canola, it's quite wild. Camelina sativa false flax is more of an emerging crop relative to canola, which is highly domesticated. So we have this kind of gradient from the wild to the highly domesticated and from evolutionary distance from Arabidopsis. And lucky for us, the Pseudomonas syringae strain, PTO DC3000, could cause disease on both Camelina and canola. As I'm showing here on the right, you see the yellow plants on the left side of the array of leaves there. They're all yellow, right? For all Arabidopsis, Camelina, and Brassica napus at the bottom. You put one ETI elicitor and immune elicitor in DC3000, and you get resistance in all three plants. So that means that we could do a screen for resistance similar to what we did in Arabidopsis. We could screen this effector diversity panel we had for resistance in false flax as well as in canola. And this was all done by a graduate student, Claire Bright McNally. First question we had was, we had 19 immune elicitors that were recognized in Arabidopsis. How many of those 19 are recognized in these oilseed crops? There's a lot in the literature about, how domestication has potentially limited the immune spectrum of crop plants. So we really wanted to test that, if that's true in this case. So when we tested all these immune elicitors, I'm showing here a bacterial growth assay. So anything that's significantly less than the gray line on each one of these graphs would be considered an immune response. It's reducing bacterial growth significantly relative to the control virulent pathogen strain. And to summarize here, we lost 1 immune response in Camelina and 4 in the more distantly related canola. But overall, the majority of immune responses were retained from Arabidopsis to false flax to canola, meaning that in this case, domestication has not substantially compromised the immunity potential of these crop plants. So now we could ask, well, okay, now these are the 19 that elicited immunity in Arabidopsis. How about our 530? How good are crops at recognizing the diversity of effectors that we studied in our Arabidopsis study? So Claire did a very similar setup that Bradley had used in Arabidopsis. So she basically screened our SciTech 530 virulence protein diverse collection in both Camelina and in canola, looking for immunity. And you can see I've highlighted in red boxes here things that would look like an immune response qualitatively. We could quantify this again with our software that monitors for yellow versus green pixels. And all these red dots would be something that are significantly reducing the disease symptoms when the immune elicit or an effector is present in the Pseudomonas syringae strain. So again, this is all done using Pseudomonas syringae, infecting on the top here, canola, on the bottom, Camelina. So to summarize this, we found 12 new immune eliciting families in canola and 17 new immune eliciting families in Camelina. And to summarize that, that means that the oilseed crops actually recognize more effectors than Arabidopsis did. So Arabidopsis, we had 19 of the 70 effector families being recognized. In Brassica, we have 29 of the 70. And in Camelina, over 50%, which is 36 out of the 70 families. And what was really intriguing is this actually correlates with the number of resistance proteins in each one of these crops. And the resistance proteins increase with ploidy. So Arabidopsis is diploid, canola is tetraploid, and Camelina is hexaploid. And the number of resistance proteins goes up with your ploidy. So does the NLR number, right, the resistance protein number, and the number of immune responses. So you get a correlation. So first of all, oilseed crops actually possess an expanded immune response relative to the wild Arabidopsis. And that immunity is proportional to the resistance or NLR proteins that are found in those crops, which is proportional to the ploidy. So by increasing ploidy, you increase NLRs, and you increase the immunodiversity, or the potential to recognize more pathogens. But what we found was when we looked at the strength of each one of those immune responses, it looked like it was getting weaker with the number that you can recognize. So what I'm showing here is basically the difference in bacterial growth for each one of these immune elicitors, whether when you have the immune elicitation versus when you don't. So basically, how much does the immune response reduce bacterial growth? So the higher on this graph you are, the more you reduce bacterial growth, meaning the stronger your immune response is, right? So what you can see is that Arabidopsis, the wild ancestor, has the strongest immune responses. It's the highest on this graph. And then it slowly goes down to Camelina, which has the weakest immune response. But if you remember, Camelina can recognize the most effectors, but overall they tend to be weaker. Which leads us to the question, does the strength of the immune response decrease with the size of the landscape or the number of strains you can recognize? Is there some kind of trade-off there where you can recognize many effectors, but somehow there's some constraint that limits the strength of those immune responses? So then we mapped everything back onto the Pseudomonas syringae phylogeny, and like Arabidopsis, Camelina and Canola can recognize almost all of the strains. But what was even more impressive was when you look at how many immune elicitors per strain, there's a lot more immune elicitors per strain if you're looking at Camelina as a host, they have the most, and then Brassica, Napus, and then Arabidopsis. All right, so you can see that on this graph. If you look, let's start with 4 or more. That's a lot of immune elicitors in one strain. How many strains have at least 4 immune elicitors? That's a lot. Well, in Arabidopsis, there's only 4% of strains that would be recognized with 4 immune elicitors. But in Canola, there's over 50% have 4 immune elicitors. And in Camelina, it's 73%. Okay? Meaning that immunity here, this ETI response, this gene-for-gene resistance is pervasive. It's like Arabidopsis. It can recognize almost all these strains. And it's heavily stacked in oilseed crops. Okay, so now I'm going to flip to the resistance genes. We got some help from Isabel Parkin to develop a ZAR1 mutant. Remember, ZAR1 was a resistance protein that can recognize many effectors by monitoring a kinase network or perturbations to a kinase network. So we asked, does a resistance protein recognize the same thing in Arabidopsis versus oilseed crops? So we knocked out ZAR1 in Canola and in Camelina, and basically what we found was only one of the 5 immune responses was lost in both of these oilseed crops. These are the 5 immune elicitors that we found in Arabidopsis. Only one of those was lost in each one of these crops. So ZAR1 is only contributing to 1 out of the 5 that were found in Arabidopsis. So meaning that only one of the 5 Arabidopsis ZAR1-mediated responses are also ZAR1-mediated in oilseed crops. But does ZAR1 recognize other things? Can it recognize a lot of effectors, just not the same ones as were found in Arabidopsis? So Claire screened all of the known ETI elicitors, and she's only done this so far in Camelina. Brassica work is ongoing. But what we're already finding is if we look at the empty vector, which is the EV with the red arrow at the bottom, anything that's different from that would be ZAR1 recognized. And you can see there's only one there that's in orange on the far right-hand side, and that's HOP-Z1A. That only one effector can be recognized by ZAR1 in these oilseed crops. Meaning that ZAR1 plays a diminished role in ETI landscape of oilseed crops relative to Arabidopsis. And importantly, the ETI landscape is conferred by convergent evolution of NLRs or these resistance proteins. All right? So that means that a resistance protein that recognizes an effector in Arabidopsis is unlikely to recognize the same effectors in oilseed crops. Okay, how can we use this to protect plants? So what I'm really excited about is developing biocontrol. And what we're doing here is on the left is canola, on the right is camelina, and we've pre-sprayed these plants with a non-pathogenic Pseudomonas strain. It has no effectors except it has a secretion system. We've pre-sprayed on the very left just that strain, and then 24 hours later, we spray with a virulent bacteria. And you can see the disease. And then on the right-hand side there, we have greener leaves. Those are the ones that have been pre-sprayed with a biocontrol that elicits effector-triggered immunity. And we see the same thing with Camelina. We seem to be able to pre-treat plants with an ETI elicitor and protect the plants. And you see greener plants and you see bigger plants. So we're really excited right now to try to develop this, to develop immune cocktails and see if we can protect crops beyond Pseudomonas syringae using these immune cocktails. Can we stack immune responses? Can it protect against blackleg, for example? Can we use different vectors to deliver these immune responses that would be more amenable to industrial acceptance? And with that, I'd like to end and thank everyone who did the work. David Gutman is a collaborator. We co-supervise everyone that's doing this work. And thank you to Isabel Parkin, Rong Zhao, and Hossein Borhan for the canola work.
Thank you, Darrell, for a great talk. So we can have one question from the floor or from online. You're really floor. Sorry. Yeah. So I think, Jane, there's a question from the...
Is there a question from the floor? Here, yeah, okay.
I'm coming with a mic. Sorry.
Got one here.
Okay, thank you.
Hi, very nice talk. So when you're talking about these ETI cocktails, one of the big things is stabilization, so you can actually use them in the field. How do you view it? Are these going to be microbes per se, isolates of the pathogen?
Yeah, so we're really excited about using growth-promoting bacteria that have type 3 secretion systems, like Pseudomonas fluorescens, for example. It's a commensal. It actually promotes the growth of plants. It has a type 3 secretion system, and it infects roots. So maybe we could do a soil drenching. We could try the spraying. We can see how different, but we're really excited about using the bugs to deliver.
Why don't you try gene editing the ETIs right into the plant?
Yeah, that would be great, but I'm not sure how what's the acceptance to that? But yes, that would be an alternative. Have the ETI elicitor that's induced by infection, and then you get an immune response, yeah.
Thanks. Thank you. Please join me in thanking Darrell for a great talk. Thank you. The next speaker of this session is Steve Strelkov. He's a professor of plant pathology at the University of Alberta. Steve's presentation entitled Clubroot of Canola Surveillance and Management. Please welcome Steve to the stage.
Thank you. Thank you, Steve. There's my timer there. And this,
I think this is about,
okay.
About 2 minutes, 30 seconds.
Okay. I'm wasting precious seconds. I saw we're down 6 seconds already. So, quite a change of focus. So, I'll be talking a bit about Clubroot, provide a bit of an update on the disease situation in Alberta and a bit of work we've been doing. Before I get going, I'd just like to acknowledge some of the people in our lab that contributed to this work, as well as Mike Harding from Alberta Agriculture and Irrigation, and Xiaofeng Huang, also in my department as collaborators. So just a brief outline. I'm just going to start off with an introduction to Clubroot. I suspect everybody here is fairly familiar with the disease, so that'll be very short. When I talk about the disease situation in 2024, present some of the more recent pathotyping work we've been doing and talk a little bit about pathotype shifts, and mention a little bit about some of the qualitative differences we've been seeing in disease development beyond just quantitative changes, and just finish off with some conclusions. So I think most people in this room are familiar with Clubroot. It's a soil-borne disease of crucifers, including canola, caused by the obligate parasite Plasmodiophora brassicae. It attacks, as the name implies, the roots and causes the formation of these large clubs or galls of the root system, which interfere with water and nutrient uptake. And when symptoms are severe, it can result in very significant yield and quality losses. Now, since I started at the University of Alberta in the fall of 2003, at the same time that the first couple cases of Clubroot were reported in the province, And so since about 2004, 2005, we've been conducting annual Clubroot surveys across the province in collaboration with municipalities as well as with the, Alberta agriculture. So in these surveys, we basically inspect crops for symptoms of clubroot, look for, we prioritize the field entrances because if clubroot is going to be found anywhere in the field, it's usually going to be found at the entrance. If the symptoms are found, we inspect the entire field as shown by that diagram on the bottom, kind of in a W transect, and we sample 10 points along the W. And we also will look at sometimes low-lying areas in the field and so on that might be more favorable to clubroot. And then we enter all this data into a Clubroot database we maintain at the University of Alberta, which has harmonized data from 2005 to 2024. And this includes the field geolocation, the year or years where Clubroot was identified in that field, whenever it's available, the host variety from which the plant was obtained. Sometimes we'll also have pH and other information, as well as the pathotype found in that field when we have that available. So for the 2024 survey, the province-wide survey visited 691 fields, and this was the University of Alberta and also Government of Alberta. And we coordinated with the counties and municipalities in all of the fields that, in all of the regions that we visited. And we tried to get, kind of representative coverage of canola acreage and also coverage of at least some of the counties in each of the 12 canola regions identified by Alberta Canola. And then we compiled all the data from all these surveillance activities. As of 2024, we were able to identify another 307 canola crops in the province that had clubroot, and the map shows the distribution kind of the disease. And where it's more reddish, there's more disease cases, it's more severe. And there were, of those 307 canola crops that had clubroot symptoms, 167 cases were found in fields that had no prior history of clubroot. So those were the first detections of clubroot in those fields. And we also had the first detections in Clear Hills County, which is up in the, I'm not sure if I have a, let's see if the pointer works, up there, it's sort of in the Peace Country. So that was, so that brought up to 47 counties and municipal districts in the province that have documented cases of clubroot. And that's 47 of 66 counties or municipal districts where canola is grown. So about more than 70% of counties in Alberta now have documented clubroot. And okay, yeah, and I just wanted, since this is not just an Alberta meeting, I wanted to compare our numbers with a little bit of what's been reported from Saskatchewan and Manitoba. So in Alberta, clubroot was first identified in 2003. And we've been able to document the disease, like symptomatic crops of canola crops in 5,833 canola crops between 2005 and 2024. And these canola crops represent 4,100 individual fields that have confirmed clubroot infestations as of 2024. So of course, in some fields, we found clubroot over multiple years, hence the number of total cases is greater than the total number of fields. But this compares quite strikingly with Saskatchewan and Manitoba. In Saskatchewan, the first case was in 2008, and as the latest that I could see was 82 fields with confirmed clubroot as of 2023, and with an additional 43 fields that tested positive for the presence of the pathogen DNA through soil testing. And now in Alberta, we typically don't do DNA testing of the soil because the disease is so prevalent. In Manitoba, it's kind of a similar situation to Saskatchewan. The disease was first detected on canola in 2009. As of cumulative cases, the most recent numbers I could find was 35 fields as of 2019, but nearly 300 fields where the pathogen DNA, Plasmodiphora brassicae DNA, has been identified in soil samples. So in Alberta, the outbreak is still like an order or 2 orders of magnitude more severe than Saskatchewan and Manitoba. Since basically we started our surveying, we've also been doing pathotype monitoring over the years. I think, again, most people here would know that clubroot exhibits physiologic specialization and consists of many different pathotypes, or sometimes called races. And in Canada, for about the last 6 or 7 years, we've been classifying isolates into pathotypes using the Canadian Clubroot differential test. And really, part of our testing is to see, track pathotype shifts and any changes in virulence. But this is quite a labor-intensive work, as you can see, based on those image of some of the greenhouse testing. So, in terms of pathotype composition from 2021 to 2023, that we have the most recent data that we just finished compiling for publication. We tested 206 field isolates. The vast majority came from Alberta. 194 of them were from Alberta that we basically picked up during our surveys. We also received 8 samples from Saskatchewan from testing and 4 from Manitoba. The majority from Alberta were resistance breaking, 3/4 of them. And I just tried to put this image because I circled it. You can see resistance-breaking pathotypes are pathotypes that can cause clubroot in at least some of the club-resistant canola varieties that are available. And in this map, we show individuals, fields that were tested. And where it's purple, it's resistance-breaking. Where it's green, it's controlled by all sorts of resistance. And what you can see is that most of the resistance breaking is occurring certainly in the central Alberta, where clubroot has been most entrenched. And as you move into, let's say, more clubroot peripheral areas, there's a lot more of the older type pathotypes that couldn't break resistance. But although some resistance breaking is beginning to emerge there. In any case, in the 2021-2023 period, we found 31 unique pathotypes, bringing up in terms of total documented pathotypes in Canada to 55. This is up from 17 in 2016 and 43 in 2020. And this suggests that there's some rapid diversification of these pathotypes or virulence patterns. Of the 55 pathotypes, 35 overcome the resistance in at least some clubroot-resistant canola, and 20 pathotypes appear to be controlled by all clubroot-resistant cultivars. The majority are rare or geographically limited, and these graphs just show kind of the composition of pathotypes. So on the top, we have a pie chart showing province-wide, and on the bottom three pie charts we have for Alberta, for Saskatchewan, for Manitoba. But keep in mind, it's not really an equal sampling. There's 194 for Alberta, while only 8 and 4 for Manitoba, 8 for Saskatchewan and 4 for Manitoba. But in any case, as in previous years, we've found that pathotypes 3 and 3D, which are resistance breaking, as long as with the original kind of non-resistance breaking pathotype 3H, remain the most common. Each represented about 15 to 25% of all isolates tested per year, and more than half of all isolates when you combine across the 3 years and you combine those 3 pathotypes, 57%. However, they're not as predominant as they once were. If you go back to 2018, 2020, when they would make up 60, 70, 80% of all pathotypes we were finding. And there's other pathotypes that we have noted are increasing. They're still fairly rare, but they're not one-offs. For example, maybe 4 to 8% of all samples tested. Potentially concerning are some pathotypes that we call kind of pathotype 9 variants, namely 9D and 9E, and I'll just mention that I'll give a bit of information on that in a couple of minutes. Now, when I've been talking about clubroot resistance breaking, very often the hosts that can become infected by clubroot are what we sometimes call first-generation resistance. Basically, canola cultivars, the first canola cultivars that had resistance to clubroot most of them seem to share a similar resistance source from the winter oilseed rape menthol from Europe. Now, with the advent of resistance-breaking pathotypes and virulent pathotypes, there was definitely an interest in developing varieties with new resistance, and sometimes these are collectively termed second-generation resistance, although we're not referring to necessarily a single resistance source. These could be different resistance genes or combinations of resistance genes, but it's kind of a catchphrase used to summarize that. So we have been doing in a project that just finished in 2023, I believe. We were looking at the behavior of some of these second-generation club-resistant canola cultivars and the resistance to isolates of Plasmodium brassicae representing different pathotypes and collected from fields that had second-generation resistant varieties grown on them. So you can see two groups of hosts. On the top, we have a check host. So each circle represents basically what proportion of the isolates each host was resistant or susceptible to. So blue means resistant, orange means susceptible. So on the top, we have the check host, which is ECDO2, which is a Brassica rapa that's been resistant to everything we've ever tested against it from Canada. And you can see it was resistant to all of the 36 isolates in this study. We have ECDO5, which is another Brassica rapa, and that's the Clubroot universal suscept. It bears absolutely no resistance to Clubroot and never has. And then we had on the right-hand side, on the top there, 45H29, which was the very first Clubroot-resistant canola variety, and that was kind of our first-generation Clubroot-resistant check. And that was resistant to about half of the isolates tested. On the bottom half, we have second-generation resistant cultivars kind of coded, just SG1 to SG7. These are maybe a bit older now because these were picked like 3 years ago, so they're not necessarily varieties that are still on the market, though some of the resistant sources may be. And what you can see is a lot of variability in how some of these second-generation cultivars were performing. Some performed very well, like SG7 there. SG3 was pretty good, SG1, and then some were actually quite poor. They actually performed worse than the very first-generation resistant variety. What we also found was that pathotypes 3A, 9D, and 9E were virulent on most hosts. And of all those tested, the isolates from 9D were the worst. They attacked, they could cause severe clubroot on all hosts that we tested except ECDO2, so except the resistant check. So that's definitely, those pathotype 9 variants are something to look out for. Right now, they're still geographically, we find them mainly between Edmonton and Calgary. That's where most of the vast majority have come. But so maybe there's other things that are restraining their advance, but certainly they seem problematic in terms of their virulence. Now, we know that specific host pathotype and host variety interactions lead to quantitative differences in clubroot severity, how severe the clubroot is. But we're also finding differences more lately now, as we've been looking into it more carefully. We've been finding qualitative differences in clubroot development. So in terms of how the disease is developing on the roots is quite distinct. And that lends itself to the question, could these differences have an epidemiological impact or maybe affect the performance in terms of how the crop can deal with clubroot? So these are just some images we create, some time lapse we did in our lab to show, just to highlight the differences in development. You can see on, I guess it would be on your left-hand side, we had a combination where the clubroot basically attacks the entire root system, including the taproot, and the whole root system becomes kind of galled. And then we have other situations where you have very severe on the left, I guess it would be on your right-hand side, where we have very severe clubroot, but it's restricted almost to the top there, to the crown, and while the root system remains, for the rest of the root system remains fairly normal. So this does show distinct development of galls on different hosts, and certainly it could have sort of epidemiological implications. So I have only 1 minute left, so I come to our conclusions. Clubroot, of course, continues to spread in Alberta. It's certainly in Saskatchewan, Manitoba, but it doesn't seem to be still spreading nearly as bad as in Alberta, where the situation is most severe. 55 pathotypes are now documented on the Canadian Clubroot differential set, and pathotypes 3A, 3D, and 3H remain predominant, but other pathotypes are increasing. And we have been finding these both qualitative and quantitative differences observed in the disease development. So not all galling, even if it's severe, is looking the same. And just wanted to acknowledge county and municipal district staff that helped with the surveys, of course, the growers and collaborators, and all the funders without whom this work wouldn't be possible. Thank you very much.
Thank you, Steve, for an interesting talk and providing an overview of the Clubroot disease. So we have time for a couple of questions, either from the audience or online.
If not, we do have one question online here, if you can hear me. Are the differences in qualitative clubroot development on roots correlated with soil moisture and other environmental factors, or are they purely genetic?
Right now, we're just starting to do some of this work. This is all being produced under the greenhouse conditions, under identical conditions. So I think it's more reflective of the interaction, genetic between the host and the pathogen, rather than between, that they're being grown under different conditions or something like that. Yeah.
We've got one more that just came in online, but is there a question from the room anywhere? No. Okay. So here's a good one online. Do you have any speculation, any ideas why there are still higher clubroot rates in Alberta compared to the other provinces?
Yeah, that's a very good question, and the fact that it's such a big difference. I guess one could hope that on the positive side, that by the time that Clubroot really got going in Saskatchewan and Alberta, in Alberta we didn't have any genetic resistance until 2009. We had very limited understanding of the pathogen in those early years, and we were already dealing with hundreds of cases. While in Saskatchewan, Manitoba, when the first cases came out, we already had, we coincided with the onset of a lot of management tools such as genetic resistance and recommendations to growers. So I think maybe that could be part of it. And of course, in Alberta, we have a much higher percentage of acidic soil, so that certainly favors the pathogen. So that could also be, a reason.
Thank you, Steve, for the informative presentation. Actually, I got a question about, like, every year we see that the pathotype or races is increasing. So last year probably it was 36, now it is 57 or so. It is getting higher and higher. So I was just wondering, what might be the cause? It's the selection pressure of new resistant variety to getting this number higher, or any reasons behind it?
I think so. I think certainly that by growing those resistant varieties, we're selecting for maybe very rare element of the population that maybe have different virulence patterns on other hosts. So I think that's probably been, and of course the continual monitoring allows more and more detection over the time. But like I said, even though there's lots, it's still dominated by a handful of pathotypes that make up the greatest number. Yeah.
Thank you. Thanks, Steve.
Okay, thank you.
Thank you. So the next talk is by Team Dumonceaux. He's in Agriculture and Agri-Food Canada in Saskatoon, and he's a research scientist. His interest is in agricultural microbial ecosystem, biofuel, and also molecular diagnostics of agricultural pathogen. Team's presentation is in use of biopesticide as a novel management strategy for Sclerotinia in canola.
Okay, thanks very much, Hossein. I really appreciate it. Appreciate the opportunity to address you all here today. And I want to begin by making an observation that's kind of intuitively obvious, but it's really rarely quantified. And that is that we inhabit a biosphere that is essentially composed of plants and microbial life. So the majority of life on Earth is composed either of plants or of microbes. And it's been this way for a very, very long time. So plants and microbes have had a long time to develop an incredibly dizzying, diverse array of interactions. And of course, the interactions that really get our attention are those that are negative interactions, the ones that result in disease. But if you really quantify the interactions between plants and microbial life that are found within the biosphere, you find that the majority of these interactions are in fact beneficial. That the majority of the time that plants and microbes interact, it is beneficial. This is a quantification here of the application of soil bacteria to the growth of plants. And you see the response of the plants in terms of biomass increase with the application of these microbial species. And you see that the median here, indicated by the red line, is above 0. So the median response of plants to the application of the microbial life with which they are surrounded is beneficial. We don't often notice it. It goes unnoticed because the plant is simply healthy. What we notice are those interactions that result in an interaction on the negative side of this line. So these considerations have led us to this concept of the holobiont. Now, this is not a new concept. This has been around for more than 100 years, but it has really seen a resurgence lately where we consider the interactions between the microbial life associated with plants as an organismal interaction. It's actually part of what we could consider a plant. A plant is not a plant without the microbial life that interacts with it. And it's easy to consider this if you go back to what defines a plant to begin with, these events that happened over 1 billion years ago where some proto-eukaryotic cell began to actually imbibe or take in these microbial species that ultimately became the organelles of the mitochondria and the chloroplast. That defines what a plant cell is. And as time wore on and plants grow in this microbial bath, both above ground and below ground, they've developed all of these interactions. This is really fundamental to life in our biosphere. And of course, some of these interactions are negative. We see this in terms of plant diseases and indeed in animal diseases. But it can also confer beneficial traits. Microbial species are found all throughout plants, on the aerial parts, on the subterranean parts, within and between plant cells, on the outside of the plant cells, all throughout the plant. And really, this is the concept that becomes this idea of biocontrol. I want to explain biocontrol as I understand it. So we have all these ranges of interaction between microbial species and plants, some of which are negative, as you see here with the crop pathogens. But in other cases, you have these beneficial microbes that will control that disease, and you don't see the disease manifesting. It often happens invisibly. We don't notice that it's actually happening. But this is going on as plants grow and develop. Now, when we were all in school, we all learned to consider this group of ecosystem activities under the kind of dry and boring term of ecosystem services. But I want to introduce a concept that is really important, really in indigenous science, as described by the author Robin Wall-Kimmerer, where she says that we can really reframe this idea of ecosystem services and consider them instead as ecosystem gifts. These are gifts that this microbial life gives to humanity by controlling these diseases. And when we reframe it in this way, when we consider these things not as services rendered, but as gifts freely given, it changes our perspective. It forces us to really think about this and to really take on a responsibility of using the tools of science to honor these gifts by studying them, by understanding them, by applying the tools of science to understand these gifts, to reveal their mechanisms, and in fact to give them on, to re-gift these gifts and use them to support human objectives of improved plant growth. So that's the framework within which I understand biocontrol. So the particular application that I'm going to be talking about of using biocontrol is a very well-known plant crop pathogen that the next two talks will talk about, so I won't spend too much time on this, But this is a fungal plant pathogen called Sclerotinia sclerotiorum. It infects, of course, canola, that's why we're here, but also many other plants of human interest. And it has a really complex, multifaceted life cycle where it can infect through the air when these apothecia germinate and produce these ascospores that float through the air, land on the plants, and start to cause disease. But when the sclerotia that are in the soil germinate, they can also infect canola and other plants through their roots. So this is a complex pathogen, causes a lot of disease and economic losses. So of course, producers have responded with control methods, and the most commonly applied and well-known would be fungicides, and they're very effective. That's why they're used. I don't need to describe the drawbacks associated with that. There's also physical and chemical control methods that have been applied, and these can be effective. And of course, the ultimate solution is the one that's going to be the subject of the talk by my colleague in the next presentation, really closing the door to the infection altogether, is breeding, a really important aspect of disease control. So we have this project that was funded by the Canola Agronomic Research Council, where our objective here was to identify and develop an effective biocontrol agent for Sclerotinia-associated stem rot in canola. So we had three specific objectives which fit into this concept of biocontrol as I've described it. The first is to identify these candidate bacteria that actually possess these ecosystem gifts. The second is to understand the mechanisms, to honor those gifts by understanding how they work. And the third is to really give them on, to develop ways that we can present these gifts to producers in a way that they can actually be applied. So how was this approach? So the source of the bacterial strains that we used is the legacy of our late colleague Susan Boyechko, who spent her career amassing a freezer full of strains, of around 2,000 or so strains isolated from various sources. And Sue had a great feel for her collection. When she had a new biocontrol challenge in front of her, she kind of knew in her head which strains she should approach and to select out of the freezer. So her initial screen is only 15 strains, and she used 3 different methods to screen out strains that might show this effect. And I'm only going to show one of these 3, and that is by producing these metabolite plates. So you grow the bacterium, you spin away the bacterial cells, and you use the cell-free culture supernatants to make some agar plates on which you grow the fungus. And those plates on which the fungus doesn't grow very well are derived from bacteria that are producing metabolites that inhibit the growth. They exhibit this phenomenon of antibiosis. So you can see here some strains have more efficacy than others. And through this screening method, Sue was able to identify a series of strains with a variety of antibiosis activities, some of which were quite effective. Ultimately, we settled on one particular strain. You can see here when we identified them taxonomically that there are mostly strains of Bacillus. Many of them were Bacillus atrophaeus. And for reasons I won't go into, we zoomed in on this one strain of Bacillus atrophaeus called PENS V20 for really exploring this biocontrol effect. So we explored PENS V20 as a biocontrol agent. So we looked at the efficacy of this bacterial strain for controlling Sclerotinia stem rot on canola. So we tested it on 2 different cultivars, Westar, which is the cultivar that's susceptible to everything, but also a partially resistant cultivar we got from our colleague Feng Chun from China called Zhongyu. This is a winter canola cultivar, requires vernalization for germination. It's a bit of a pain to work with. But you can see here that the middle plants have been sprayed with the biocontrol agent plus the pathogen. The plants on your left are sprayed with pathogen only, and they're effectively dead, and compared to the plants on the right, which were not challenged with pathogen. So we think we've demonstrated here that PENS V20 does demonstrate this strong protection versus Sclerotinia under these really favorable conditions. You can see how devastating the disease is. But of course, we didn't just, I'm going to show you the pictures of the best ones. We did this with scientific rigor by doing the experiment 3 times in a fully randomized greenhouse trial, and these are the results. The stars here indicate statistically significant differences as compared to the water control untreated with pathogen. And you can see that amassing these 3 biological replicates together, that of course The plants sprayed with pathogen only were obviously different from the water control, but many of the biocontrol strain treated regimens were actually not distinguishable from the control. We also observed greater plant growth and less disease in West Star compared to Zhangyu. So to look into the mechanisms of this, we had our colleague Chris Kirby at Charlottetown examine the active metabolites that are being produced. And to do this, he grew the cultures in standard lab growth medium, separated out the metabolites, and then applied them to TLC plates, which separates these metabolites out, sprayed those plates with mycelial suspension of Sclerotinia and grew them out. Those metabolites that are inhibiting the growth will show a halo of non-growth, and these can be identified. And this is what it actually looks like. Of course, the cartoon always looks better. But you can see here that there are metabolites produced by all of these strains that will actively inhibit the growth of Sclerotinia. And he identified these as these lipopeptides, the classes iturins, surfactants, and fungicins. These were already known to be lipopeptides with antifungal properties. So this was encouraging that these are the metabolites that are being produced. So we sequenced the genome of this organism to identify regions that might be associated with the production of these kinds of antifungal metabolites. And we identified all kinds of different regions, including those that are producing the metabolites that we detected. And we can actually localize them within the genome, and we can see that these are some of the mechanisms by which PENS V20 can actively inhibit the growth of Sclerotinia. And then we also looked at the plant-associated bacterial database to really look at genes that are associated with pathways that are part of being plant-associated and inhibiting the growth of pathogens. And you probably can't see it very well, but one of the things that stands out here is that there's a lot of pathways associated with the induction of systemic resistance, for example, which is another aspect that we're looking at. Biofertilization, so helping the plant to access minerals and nutrients in the soil, and other things that are associated with plant root colonization and the utilization of plant-derived substrates. So we see all kinds of different mechanisms that this plant uses to associate with plants and to inhibit the growth of the fungal pathogen. So that's kind of using science to reveal the mechanisms. Now we want to think about how we can repackage this gift and give it on to producers. And that's by formulation of this Bacillus atrophaeus in a way that it could be applied actually to plants, not growing it in a laboratory growth medium. So our objective here was to make this strain available to producers as a viable option. And by viable, I don't mean biologically viable. That's just obvious. I mean economically viable, because without that, it will never become available. So we have to consider things like keeping the costs down while leaving room for a bit of a margin so that this can actually become available. So we worked with an established company in the United States called Montana Bioagriculture that has experience in the formulation and application of these things. And Cliff at MBA provided a series of different formulations. As you can see, we're starting here with formulation 11, so there was a little bit of playing around with these to get the conditions just right. And they come as this sort of wettable powder, and that powder contains viable bacteria that can be suspended into can be put into a suspension and sprayed onto plants. And when we did this in the greenhouse in the same manner as when we grew the biocontrol agent in laboratory growth medium, we see the same sort of thing. We're growing these plants in conditions that are very favorable for the development of disease. So you can see when we spray with pathogen only, the plants are sticks after a few weeks of growth, whereas the plants that are protected with all of the formulations, essentially, as you'll see, they are really difficult to distinguish from those water controls that have not been sprayed with pathogen at all. And you can see that the leaves looking a little bit white. That's a little bit of a chalky residue that's from the formulation itself. It didn't really seem to bother the plants too much. But again, when we went through and we did this with a fully replicated trial in the greenhouse, and we compared each of the groups, this is a sort of a disease severity rating that's kind of a whole plant assessment of how much disease we're seeing on these plants over time, so days 7, 14, and 21. You can see that the only statistically significant difference from the water control was the pathogen only. All of the formulations really effectively controlled disease, as did the group there, BCA+1 is our biocontrol agent just grown in a laboratory growth medium. So we think that this was quite effective, but it's a little disturbing to see that there was some disease severity that was noted even in the water control not sprayed with pathogen. I suspect that that could be just non-Sclerotinia molds. These plants were very, very wet to keep the disease conditions very amenable. So it's easy to mistake that when doing the ratings. We have some molecular data that suggests that those bars are effectively at 0 for Sclerotinia itself. But where we did see some differences, though, with the formulations is in terms of plant growth, as reflected in the weight of the plant or the amount of biomass that's produced at the end of the experiment. And on the left here, we see the water control that's kind of set as our standard. All of the others compared statistically to that. And you can see here that some of the formulations, this brown bar here, formulation number 11, even though it controlled disease pretty well, still the plant didn't quite do as well as with water control. But 2 of the formulations, in particular 14 and 17, were really indistinguishable from the water control. And the pathogen, of course, those little sticks there, they didn't weigh very much. The amount of biomass was very low. And our colleague Steve Robinson actually applied a statistical method called beta regression to look at these results and to use them to sort of predict what might happen if we were to repeat this experiment. And what we see here is a projection of what the disease severity might be if we repeated this experiment. And you can see here that all of the formulations were difficult to distinguish from the untreated water control. The pathogen was experienced severe disease. But some of the formulations were slightly better than others. You can see that formulation 11 was slightly to the right there towards the increased severity, whereas all of the other formulations, in particular 14, 16, and 17, were even better than when we just grew the strain in laboratory growth medium. So to conclude, we identified many strains that feature this phenomenon of antibiosis. Most of them were bacillus strains. Several were Bacillus atrophaeus. We've identified a primary biocontrol candidate. We've looked at the mechanisms of action, and I've only gone over it very quickly. I didn't talk about the stimulation of host gene response, which is something else that we've looked at. And then industrial scale formulations are effective at biocontrol, and we've demonstrated that. And our next step is the really challenging one, of course, which is field trials. And we've formed a bioproducts partnership now with Montana BioAg, which is now called Monterra Canada. So it has opened a branch here in Canada to really bring these things to field trial and try and bring these ecosystem gifts into the hands of producers to re-gift them in that sense. So I'll end there, and you can see that there's a lot of people that were involved in this, but I really have to mention 2. And one is the Canola Agronomic Research Council, which supported the work financially, unflaggingly through all the challenges of COVID, through the unexpected death of the project lead, Sue Boyechko. And of course, I have to mention Sue, whose vision this was, and who it's our pleasure and our privilege to try and bring this vision through to reality. Thank you very much.
Thank you, Tim, for a very interesting presentation. So we have time for a couple of questions.
Wrong direction.
Very nice talk, thank you. Since it's a bacillus, have you thought about using it to apply to the soil as a seed treatment? And then with your current testing, how many applications do you have to make per season to get the control that you would expect to see under field conditions?
Yeah, that's a great point. At the way we're currently doing it is greenhouse only, and it's a single application. And I didn't quite emphasize it, but it's either on the same day as the ascospore application, the day before, or the day after. We found that the most effective was the day after, but a single application. Under field conditions, that's for the next stage of the study. And as far as seed treatments, absolutely we're considering that with this and with other biocontrol agents, particularly those that show plant growth promotion.
Thank you for your nice presentation. Actually, my question was, I got your answer. How you inoculated your plant? Is it ascospores or mycelial
Ascospores.
Ascospores, okay.
If I had time to show it, I could show Jennifer, the technician. She did an enormous amount of work collecting these ascospores.
So what was the concentration?
10 to the 5th ascospores per milliliter. So we just sprayed them to run off. We hit them pretty hard.
Okay, thanks.
Thank you, Tim.
Are we out of time? There were a couple on the Slido as well. If
I think, yeah, one minute, yeah.
okay, we'll pick one. What are the storage and handling requirements of the biocontrol agent? That's the first one.
Okay, that's a great question. We've been storing them now for months at 17 degrees, so it's not quite 4 degrees, and checking the viability by plate counts. It has not dropped significantly, not by more than an order of magnitudes from March until last month.